Welcome everybody. Today we have Dr. Nicholas Johnson, who treats adults and children with both common and rare neuromuscular conditions, yet his work doesn't end in the clinic. He dedicates significant time each week to laboratory research and is part of the team at VCU Health working to advance the treatment of genetic muscle disorders with a special emphasis on muscular dystrophies. Dr. Johnson is board certified in neurology, neuromuscular medicine, and neuromuscular pathology by the American Academy of Neurology and serves on its government relations committee. Among many other DM-related projects and research, Dr. Johnson was the PI of the recent Myotonic Dystrophy prevalence study and is currently the co-PI of the DM1 Study, establishing biomarkers and clinical endpoints in myotonic dystrophy type 1, a non-interventional natural history study designed and run by the Myotonic Dystrophy Clinical Research Network, a network of medical aims to support future clinical trials of potential therapies for DM1 through the generation of evidence around endpoint measures and testing methods. Thank you, Dr. Nicholas Johnson, for spending your time with us today, and we look forward to hearing from you.
Great, thank you, and thanks for having me, and thanks for sticking with the conference. I think it's been a great conference so far, and you know, I know many of you have been bouncing around. I want to really commend the Myotonic Dystrophy Foundation for doing such an amazing job putting on this virtual meeting. It's gone, I think, quite well. So I believe I am the culmination of those of you who have been listening to the research tracks. So you've heard from my colleagues, Drs. Wang and Berglund, about you know, some of the background of myotonic dystrophy and what some different approaches might be for treating these really serious disorders. And so I'm going to speak with you about the next step. What I think most of us think would be the best step or the coolest step, which is, you know, clinical trials using therapeutic agents to treat the disease. So these are my grants and disclosures. So I like to make sure we're all on the same page before we get to, you know, get into the meat of it here. And so I think it's important to think about, how do you even participate in research? For those of you who have visited the various exhibitor booths, you'll see that many people are asking you to do different studies, and that's great, and it's a good way of getting involved. If you haven't visited the exhibitor booths, or you're not sure if you want to just wait until this meeting occurs once a year, you can always go look for studies on clinicaltrials.gov. So if you go to clinicaltrials.gov and you search myotonic dystrophy, you'll see a bunch of different studies. This includes information about the age of those who would be eligible, which subtypes of DM1 or DM2, and what type of functional status. So in other words, do you need to be able to walk? Can you be bound in a wheelchair, et cetera. Those studies will include contact information. If you're not sure about the study or risks, or if you qualify, just ask. That's what that contact person is there for. So please do make an effort to continue to stay abreast of clinical trial and clinical research opportunities, because this is really a partnership between researchers like myself and those of you in the audience who have myotonic dystrophy. So I think this is going to be a question. I'm going to put the slide back up. Hopefully we talk about this again next year or the year after. What study should I participate in? That's a great question. So the good news is, if it's a natural history study or non-interventional study, so there's no drug involved, you can probably do almost all of them. So, and I think most of us would encourage you to participate in at least more than one natural history study if you do qualify. Hopefully you see a neuromuscular expert who can certainly help guide you on the best studies for you. And then, especially not now, but in the near future, drug trials do include more risk. And so you want to discuss with the investigator what those are and if they're acceptable to you. So I'm hopeful, as I'll say later on, that we'll have decisions and multiple different opportunities for drug trials for people to participate in if they're interested. So why do a natural history study? I get asked this question sometimes. Why do so many? I think we're on maybe our third or fourth iteration of a natural history study for myotonic dystrophy type 1. Well, natural history studies as a whole are a broad category. So some study different patient groups or different outcome measures, different biomarkers. And at the end of the day, the key point is, what is the goal and objective? Is the goal to follow a large cohort of individuals to describe the prevalence? Is the goal to, prepare for clinical trials? Is the goal to describe the natural history? Some of those things are overlapping, but not all of them. And so, I think that's the reason why I think we say, well, it's a natural history study. You just need one. But in fact, oftentimes, different studies have different goals and objectives, and they really kind of overlap and overline. And then the last thing I'll say about it is that, in fact, oftentimes we'll start trying to develop clinical endpoints, and we'll realize that we need to refine that, and we can do a better job. And so sometimes, it takes multiple different iterations of that study to be successful. So most importantly, these do develop tools for clinical trials, which is the objective here. So those tools are called outcome measures. So I think a lot about outcome measures. These are tools that we can use to tell if myotonic dystrophy has changed between 2 points of time. So they can be something that we measure in the blood or muscle, which is at that point called a biomarker, or they can be something a physical therapist or clinical evaluator assesses, like strength or the ability to do tasks, or they can be questionnaires that you fill out to describe your experience with myotonic dystrophy. So perfect outcome measure for us, it's pertinent to myotonic dystrophy. It's reliable. In other words, if we measure day 1 and day 2, it looks the same. This is actually really going to be a key point actually moving forward is it sensitive to disease progression? Myotonic dystrophies as a whole are slowly progressive. They are progressive, which is unfortunate, but they are slowly progressive. And so getting to an outcome measure that can measure enough change during the course of a typical clinical trial can be somewhat challenging. And then, inexpensive, there's limited budgets for some of these studies. And then lastly, feasible. So, I think many people like us here at Virginia Commonwealth University or other myotonic dystrophy research network sites have had a long history of experience looking at this, but, is it feasible to collect at multiple different sites, or including sites who are less familiar? So we haven't quite settled on the right mix of outcome measures just yet. And so that's because we need the right tool for the right job. So, for example, if we measure gene expression, that's not going to tell you whether or not somebody has pain. And different tools are important for different times in the drug development process. So you really need more than one tool to build a house. We need the clinical trial toolbox. And so, that's just how we think about it. So we think about a combination of the biomarkers and the questionnaires and the measures of physical function kind of all coming together. Some are more important at different phases of drug development. So what biomarkers do we think about in myotonic dystrophy? And this slide here is for myotonic dystrophy type 1. I think, there's ongoing work, as Dr. Hamill spoke about earlier today, about trying to replicate some of this work in myotonic dystrophy type 2. But this is a slide here of what is a great biomarker or potentially a great biomarker in myotonic dystrophy type 1. So on the right-hand side, you can see different people. Each dot is an individual person, and you can see that there are different levels of strength in those different individuals. While I don't expect you to follow along unnecessarily with the graphs above it, the gels right above those dots on the right-hand side. But what you can see is that your level of RNA splicing changes associated with myotonic dystrophy varies and is directly related to the level of weakness that can be seen, for example, in your ankle strength. And so this is a great clue for a muscle-based biomarker that would really tell us whether or not a drug treatment would be beneficial for myotonic dystrophy. And for those of you who participated in, for example, DM1 or other studies like it, this is the reason why we pay so much attention to the muscle biopsies that we perform. So as I was going to, on the left-hand side, just to get back to the point, for gene therapy, the biomarker is expression of missing protein. So this is what did for myotonic dystrophy, but for for Duchenne muscular dystrophy, but for myotonic dystrophy, it's likely to be more complicated than that because in myotonic dystrophy, you're not missing a gene. In fact, if anything, you really have a toxic overexpression of a particular part of the gene. So that's why I think RNA splicing changes in the tissue like muscle is going to be really kind of a key point in something that we're going to be thinking about quite a bit, especially for those early phase clinical trials. Looks great, but there's still a lot of work to be done. So we need to make sure those biomarkers are reliable and that they're responsible to treatment. And then we need to understand what a change in a biomarker would mean clinically in the future. And so one of the things that a researcher like myself would speculate is, okay, so if you look at the graph on the right, as I said, and could show that you could change RNA splicing a little bit, you might be able to improve somebody's strength, in their ankle. But we need to make sure that that relationship actually works, not just kind of observationally, but certainly following treatment. So we have lots of different, so those are biomarkers. Let's talk a little bit about how we think about strength and function measures. So we have lots of different techniques for measuring strength. So something called manual muscle testing, which is the picture up on the top right corner, where you can see this is what typically happens for those of you who see a neurologist in clinic. This is pushing and pulling on your muscles. We can use quantitative myometry, which is the bottom two graphs on the right, either using a fixed apparatus or there's a handheld version of the same thing. Or we can try and look more dynamically at how strength changes during movement. And so, these are some of the typical ways that we think about measuring strength. There are lots of different ways for measuring function. So, how do you walk? How do you get up from a chair? How do you go upstairs? How far can you reach? And this is a picture of one of the more clever ways that people measure function, which is, how well can you raise your arms and move them around using during the course of a video game. And lastly, we can look to try to combine some of those tasks into what we call a composite outcome measure, which tries to collect multiple different functional tasks into a single score. So there's lots of different ways to measure strength and function. Not unlike our work that we're doing in biomarkers, there's still more work to do in strength and functional measures. So standardizing the equipment and procedures, training the people who measure that, and then making sure we understand how those change with age or gender or how somebody starts at the beginning of the study. And at the end of the day, that is the key challenge for those of us in terms of clinical trial design in myotonic dystrophy. For those of you who have seen others during the conference and certainly within your family and community, you'll recognize that myotonic dystrophy, both types 1 and 2, are quite heterogeneous. In other words, two people with the same number of repeats and the same age, in the same age of onset, can look completely different, which is okay. I mean, that's part of the disease. But one of the things that during clinical trials, actually, we want to reduce as much of that variability as possible. And so, I think that's a key challenge. The second challenge being, we want to make sure that we can do the outcome measures reliably. And then probably the last challenge is shown here in the graph on the right, which is a kind of a stylized way of thinking about how clinical outcome measures might work in different groups of people with myotonic dystrophy. So you can see that the kind of light green at the bottom, the low measurement, so to speak, that's really not ideal for measuring change during the course of a clinical trial, because frankly, it doesn't change very much and it's not particularly impacted. However, if you look at the very top one, the high measure, the kind of pink one at the top there, you can see that particular outcome measure and that person may change too much. So if they're going to get to the very top of the outcome measure over 6 months, then that may be too much and not really what we're looking for during the course of a clinical trial. Most of us are thinking about things in the kind of dark green midpoint. So these are changes that we can see over the course of 12 months. They are still, fairly significant changes, but they're not so significant that we won't see the effect of the drug because the change is too rapid. So that's probably that's a major issue for clinical trial design, something that we're working carefully to try and understand and correct for. So what are the gaps left? So better predicting the rate of progression by prior performance. So in other words, if you walk down the street in 5 seconds, today, does that mean that you're going to walk down the street, in 6 seconds in 12 months? Or what if it is that, certain groups of people perform differently based on how they start on that particular test? Optimizing biomarker collection, so making sure that we collect muscle biopsies well and don't waste them, make sure that they're, they're showing what we want them to show. And then, there's an open possibility that there are other things that may help us predict whether or not someone's going to be more or less severely affected by myotonic dystrophy. And frankly, the approaching therapeutic trials has provided some urgency to these goals. So I am here with many of the other people that you've heard speak today and yesterday as part of the Myotonic Dystrophy Research Network, which is founded at the University of Rochester by Charles Thornton and Dick Moxley. And you can see at this point now that there are a number of sites really all across the United States, in Europe, that are aiming to perform this work and help us close those gaps and prepare for clinical trials. There is an ongoing study right now called DM1 that is looking for 700 people with myotonic dystrophy type 1 at the 16 centers, and you can see that it's supported by really an amazing group of sponsors, including the Myotonic Dystrophy Foundation and some of our industry partners. And the focus is really to address really, frankly, most of the gaps that I just outlined. So focus on trying to understand those outcome assessments to help us refine trial design, and those visits take approximately 4 to 6 hours. It's not that burdensome. Take a second here. If you want to participate in this study, there's my email address. You can contact me, or you can contact one of the two project managers, Brittany Holmberg. She's got her cell phone and email there. If you email me and tell me you're interested in doing that, we will get you to the closest open site. So we really do need your help participating in that study. What about children with myotonic dystrophy? I think that's has many of the same issues and gaps, and would benefit from lots of the same types of approaches. And there are now 3.5 sites. Stanford with Dr. Day's team is in the process of getting set up, and so 4 sites. You can see them there, both on the map and on the list there. And here, this study is called Treat CDM. It's a 100-patient study for children with congenital myotonic dystrophy. And these visits, unfortunately, do take more time. They take about 12 hours over 2 days. Not 12 hours each day, but 12 hours split over 2 days. and similarly focus on understanding the disease progression. And same thing, there's my email address. You can certainly email me or the project manager, Brittany Holmberg, if you're interested in participating. We will also get you the same idea, closest open site. So very much appreciate your support and help enrolling in these studies, helping get us ready for the drug approval process. So first, the bad news. It takes on average about 12 years from a drug to kind of be conceived to be approved, which is actually, it's a long time. And then out of 5,000 drugs that are conceived and tested in preclinical studies, so that means before they're tested in humans, only 5 of those drugs make it to clinical trials. And then only 1 of those 5 drugs make it to market. And the way that clinical trials work traditionally is what you can see on the top with the blue, purple, and yellow arrows. So traditionally people do a phase 1 study, which is followed then by a phase 2 study and then a phase 3 study. We'll talk a little bit about what that means and what those terms mean. But in myotonic dystrophy, both type 1 and type 2, we really, as I'll point out, benefit a lot from some of the accelerated pathways that the FDA has created for rare diseases. So, I'll talk about that in a second, but we're not on the top, we're really more on this bottom red area here, which is designed to speed the drug approval process. So, so drug approval is different for rare diseases like myotonic dystrophy. So the Orphan Drug Act, which was passed in the early 1980s by the Senate, Senator Hatch and others, provides several forms of regulatory relief. So it talks about something called a breakthrough designation and also priority review. And I'm not going to go through these terms, but really the bottom line is that drug development in myotonic dystrophy is likely faster than standard drug development for something like hypertension or diabetes. So who participates in what study? This is a kind of a good way of thinking about it. So phase 1 studies, and you might hear people start to talk about this as we talk about clinical trials. Phase 1 studies, the objective of phase 1 studies are healthy volunteers or typically mildly affected individuals. Phase 2 studies, here the focus now is not just on safety, but also what's the right dose. And so now you're looking at people who are mildly to moderately affected with a condition. And phase 3 is efficacy. So does the drug work? So it could be the same as phase 2, could be the same population. It's typically more people than in a phase 2 study. But as I'll talk about in the next few slides, some of these different definitions have been blurred a little bit more in more recent years, especially with those programs in rare diseases, because we don't have time. You can see that the graph on the bottom, which is from a cancer say, where you can see that the phase 3 talks about years to decades, and you can see there's lots of people involved, and there's just challenges in doing that in rare diseases like myotonic dystrophy. So, but it's still important to remember the phases, especially when you're thinking about which clinical trial to participate in. So phase 1 is all about safety. Is a drug safe? Phase 2, what's the right dose? And phase 3, does the drug work? This is, if you're going to take anything away, into your Saturday evening to celebrate, it's this graph right here, which is a little old. It's as of July 2019, and you don't have to pay attention to, the different companies or the phases, but the bottom line is there is at least 15 different companies with myotonic dystrophy programs, which is just amazing. That's that's something that, like I said, should give you a little bit of a skip in your step over the rest of this weekend, because what that means is that there are lots of people diligently and carefully thinking about myotonic dystrophy, thinking about how to treat it. As Eric Wayne talked about, there's lots of different ways that people are thinking about treating it. And so with that comes lots of opportunity to develop disease-modifying therapies or drugs that might slow or change the progression of myotonic dystrophy. So like I said, there's multiple different types of therapies with different cellular targets, different ways to give them. So in other words, is it a pill? Is it a injection? Is it an IV? Is it given through a lumbar puncture or other things? So there's different dosing mechanisms. And with that, we're not going to talk about it, but there's also likely different side effects with these different types of therapies. So many of those side effects and kind of the adverse reactions, we don't know yet because they're not really in human clinical trials yet. So I'm going to go back. I did this before, and I'll bring it back again. So what study should I participate in? Well, the short answer right now is that this question is just a little bit premature because, we're not quite there, but people should maybe start to think about this. So with all those different drugs, some of them are likely to address different parts or different symptoms of myotonic dystrophy. Some of those drugs are really designed to help with the muscle weakness. Others are designed to help with the myotonia or that muscle stiffness that lots of folks have. Some of those drugs are really targeting daytime sleepiness or fatigue or lots of other things. So those are just some of the examples. So first question you should ask, what's the drug targeting? What's it designed to do? Is it better for getting into my muscle versus my brain, et cetera? Second question is, do you qualify? These are the inclusion-exclusion criteria. So, are you the right age to participate in a study? Do you have the right kind of, a lot of these studies are designed to test different subgroups of population. So, can you walk or not walk, depending on the study? What are the risks? So different therapies are likely to have different risks. And then how involved is the study? Just rule of thumb, phase 1 and 2 studies, so earlier phase studies are asking more out of people who would participate in them. Typically, because they're looking at safety, there's more blood draws and there's more kind of follow-up visits. Phase 3 studies typically tend to be a little bit less involved and the visits tend to be more spread out. I would certainly, these are just hypotheticals because we're not quite there yet, but really your neuromuscular expert may be able to guide you the best studies that are just for you. So what might the drug approval process look like in myotonic dystrophy? Well, it depends. That's the short answer. I think we're going to have some time for questions here. And so I'm likely to say it depends to at least one of those questions. But it depends on the type of therapy. So is it viral vector or gene-based therapy versus what's called a small molecule? That's something like a traditional drug like Tylenol or that kind of thing. Those approval pathways are going to be different. There are different numbers of patients involved. They're going to have different timelines involved. What the safety profile is. So if a drug starts to be tested in animal studies or even in early human studies, it looks like it has a very safe drug that tends to kind of accelerate its approval pathway. And then, different studies are designed with different what we call primary endpoints. And so, if the study is designed to see how well people walk over the course of 12 months, well, that study is going to take a longer time versus a study that's really trying to target something that you can see in 3 or 6 months. So that affects the length of the study. The effect size. So, I like to use a baseball analogy here. So, I would love for every single one of those therapies that I showed on the screen before to hit a home run. But I think the reality of the situation from other disorders that we know in the past is that that's unlikely to be true. And so, if the drug ends up hitting a single or double, I think most of us in the room would think that that's still a pretty big deal compared to what we have now. But it does change how long the studies might need to be and how many different studies they might need to do to get approved for the general population. Now, if the drug starts to get tested in humans and hits it out of the park as a home run, you need fewer patients. You need to do less studies because the benefits are more obvious. Basically, it's the easiest way of thinking about it. And then a lot of this goes into what I call program design. And so different drug companies or pharmaceutical partners have been thinking about it. I mean, no company is the same. Different companies are thinking about how they want to test the drug and what populations. And so, some of that approval process likely looks different depending which company you're talking to. At the end of the day, this is a highly dynamic process. I think, most people are pretty enthusiastic when we start clinical trials. And then, as we get through the end of phase 1 or phase 2, I think that just makes a difference in terms of, does it look like it's going to be more of a single versus a home run, that kind of thing. So the drug approval process is likely to shift even during the course of the clinical trials. The good news is I think many of our industry partners are really thinking about an aggressive approach to try and bring these therapies to patients in the clinic and outside clinical trials as early as possible, which is what we want to do. I think if it's safe and it's effective and we can show that as quickly as possible, that's great because that gets it out of clinical trials and into your clinic. Can you get the therapy outside of a clinical trial? Well, as I said, this is a bit premature because there's not a lot of development programs that are out in human clinical trials, but the short answer is probably not. So early clinical trials focus on safety, and so it's really important that that's tested in the right population and in the course of a clinical trial because there are certainly risks involved. Occasionally there are open access studies that parallel later studies, but if the trials are shorter in myotonic dystrophy, there may be less of an opportunity for that. So I think it depends, but I wouldn't plan on, especially early on when the first clinical trials are being started. And then, if talking about research and clinical trials, I can't not talk about COVID-19. So nearly all universities halted in-person research visits late this spring and early summer across the United States and Europe. Some are still not quite open yet. And so there has been a delay in in-person studies for myotonic dystrophy. And additional travel bans or restrictions have created a lot of new challenges for those of us trying to perform the natural history studies, prepare for clinical trials. And it's certainly something that folks planning clinical trials or drug trials are thinking about as well. So there's definitely an impact of COVID-19 in terms of our drug development process. And it's raised new questions for us. So can we do this virtually? Can you, can you do a research visit the same as what we're doing right now and speaking to each other remotely? We need to do just the same work to think about remote visits as we do with in-person visits. Before it's kind of the old adage, you need to walk before you can run. We need to establish reliability of these virtual visits. We need to think carefully about what that looks like in patients and how to perform it safely. And so within the Myotonic Dystrophy Research Network, we're thinking and working on virtual pilots. We can still perform surveys probably. We can still probably measure function. We probably can't collect biomarkers. And then this is just my own personal opinion, but I don't believe that this is clearly affecting pharma development plans. I think we were fortunate in some ways that we weren't doing disease-modifying trials when all of the shutdowns happened, because that would very much impact the ability to test and carry out those studies. So in some ways, the fact that these trials I expect to start within 12 to 24, 36 months, I think speaks to, in some ways, it's a little bit of a benefit here, because it's been an issue for other conditions where they're trying to do disease-modifying trials. So what can you do now? Well, you can enroll in those natural histories I talked about to help us design better clinical trials. You can participate in registries like the Myotonic Dystrophy Foundation's registry and the University of Rochester's NIH registry for myotonic dystrophy patients and family members. And you can stay informed on pharmaceutical development programs. So you can do that by following along on those companies' web pages. You can follow along with the MDF newsletter and communications. And for those of you, particularly in the investigators in the Myotonic Dystrophy Research Network, many of us stay quite abreast on where these trials are and can certainly provide that information kind of in the real time. So this is just the beginning. You know, I think lots of excitement about getting started in clinical trials. But it's important to think about the end of clinical trials. And I know that seems kind of silly because we haven't really even started. But many clinical trial populations are narrower than the general population. So I was just speaking to the idea that, you know, that we, when we're trying to design a study, we're trying to find a group of patients who look more similar than not and tend to have the same kind of rates of disease progression because it allows for the appropriate study of that drug to see whether or not it works. But the counter to that is that, you know, sometimes those trial populations don't include people who are earlier or later in disease or have different kind of features. And so, you know, one of the things that we've seen in other neuromuscular conditions is that once the drug's approved, we need to work with insurers on payment models. We need to make sure that you know, that the drug is covered and that everybody with myotonic dystrophy has access to those therapies. And then, you know, even beyond that, I think, you know, given the number of different companies that have look like they have viable drug development programs in myotonic dystrophy, I think, you know, there's every opportunity to start to even think about, you know, if there's multiple drugs that are approved, can we think about combination therapies? It's not just, you know, one size fits all. So lots of exciting promise and hope, but we are just at the starting line now. And so I think I'd encourage us to pace ourselves. It's very difficult for me to pace myself. So I'm sure that many of you feel the same way. So, you know, to wrap it up, natural history studies are needs to design the best clinical trials. There is a significant likelihood of drug trials in the near future, but trial participation is likely going to be dependent on the specific design of that trial. And thank you. There's my email address again. Please feel free to email me with any questions, or if you were interested in participating in one of those natural history studies. Like I said, thank you very much for your attention. And now I'm going to try to answer questions. I'm looking at the chat function here, and I can see one question is, my symptoms change from day to day depending on stress levels, diet, activity levels, day before, and you're not the, and this person's guessing she's not the only one. That's true. And has that been an issue for research and/or setting up clinical trials? Absolutely, absolutely. So when I talked about finding outcome measures that are reliable and don't change, that's what I'm talking about. So things like fatigue, you know, I think most of us, if we think about it, you know, or daytime sleepiness, that can change a lot day to day. So that's going to be challenging to use as an outcome measure. And so, you know, measuring something like how fast you walk, that's less likely to change day to day. So we think about that a lot. Next question was, are there any studies being done specifically for myotonic dystrophy type 2? And was there ever study done before pertaining to myotonic dystrophy and gamma globulin? There has not, to my knowledge, been a study looking at gamma globulin and myotonic dystrophy. Based on the mechanism of disease, that was probably not going to be particularly helpful in terms of measuring disease progression. That question and the next individual didn't see any companies focusing on myotonic dystrophy type 2. And has there any efforts for myotonic dystrophy type 2? Well, there's a long answer and a short answer to that. So I'll start with the short answer, which is that at the end of the day, the basic biology for myotonic dystrophy type 1 and type 2, which is you have a repeat that's expanded, it binds to RNA binding proteins, and that affects RNA splicing. And so lots of the drugs that are being developed for myotonic dystrophy type 1 are designed to deal with that impact. And it's my strong belief that if any of those are proven to be effective or tractable, the very same type of approach would be likely to be quite beneficial for myotonic dystrophy type 2. So I think myotonic dystrophy type 2 is probably realistically 5 years behind myotonic dystrophy type 1. But I think it should catch up because the same basic approaches should be effective. So if we start to get a hit or an idea of a hit, we should expect to see expect to see some benefit there. So, and I know the University of Rochester and VCU will be participating in a natural history study again to try and get us up to speed with myotonic dystrophy type 2. How do you need to participate in clinical trials? Well, that depends. It depends on the clinical trial. So there are, as I said, we have a natural history study for children who were born with myotonic dystrophy and symptoms at onset. At birth, that goes up until 15 years of age, and then there's other studies that really take all comers over the age of 18. So I think I would just continue to stay apprised of these. So has COVID-19 impacted the DM1 study? Yes, absolutely. We're about 6 months behind where we want to be in terms of that. I think we have just over 120 patients enrolled so far, so we certainly need your help to get over our hump and get to 700 patients. In terms of initial data from the study released, if we come back again next year or they ask us to speak again, we're happy to talk about some of the data that we're seeing coming out of that study. Any therapeutics with drug repositioning, what I think maybe is repurposed therapeutics. There's a couple different drugs that look like they might have benefit, in myotonic dystrophy after being tested, or some of them are even FDA approved. But we need to go through the same process with those drugs, and they're sitting actually about the same place where they need to be tested in smaller groups to see whether or not they're going to be safe and what the dose is in myotonic dystrophy. Would natural history study outcomes have a big impact on biomarkers for clinical trials with myotonic dystrophy? The answer is yes. So just as much care and rigor needs to be put into figuring out how to collect those biomarkers as measuring strength and function and collecting those questionnaires, and the work that's being done in NDM1 is designed to kind of refine those biomarkers and get them so that they're working effectively. Are there any medical devices that can be worn at home? So great question to collect relevant outcome measures. We, within the Myotonic Dystrophy Research Network, are thinking carefully and have been testing actigraphy. There have been a few studies, particularly I'm thinking of one out of the London group, Chris Turner's group, that has that was measuring actigraphy, which is it's a device you wear at home for like 7 days and just measures how much you move. So those are kind of the things that that people are thinking about. Thinking about in terms of measuring function at home. So we're hoping to make it more part of the armamentarium. I think for those of us who were trying to do research studies and everything got shut down, I think it's a wake-up call. We need to be thinking differently and bigger and trying to make it more practical and getting to you in your home, just the way we're doing today. Research in herbal remedies. Yep, there has been some work and effort into looking at whether or not certain herbal remedies could be beneficial for myotonic dystrophy. The same kind of work with the drug repurposing. And so I think, that's coming as well. So there are so most natural history studies have an age window to participate in. So there's a question about restrictions on being too old. It is our expectation that early clinical trials may not include people who are, based on past experience, other diseases, there's obviously it's not set in stone, but may not include people over the age of 65 or 70. And so our natural history study, myotonic dystrophy, and DM1 has the same age cap. Is there travel assistance for patients when participate in natural history studies in states without centers? Each natural history study or clinical trial will have its own rules about travel and travel reimbursement. I speak personally for myself that in our pediatric study we try and reimburse as much of the travel as possible, and in our adult study we're adding in a little bit of travel reimbursement. So. And then we're also trying to open sites across different areas of the United States where we can see gaps on the map. Any studies for patients from other countries, and this is somebody from Australia. So the DM1 study, by the end of this fall, we'll be enrolling patients across Europe as well. I'm not well aware right now if there's any studies in Australia, but I think that would be something to take up with your neuromuscular specialist if possible. What, if any, have the effects of CBD or marijuana research on myotonic dystrophy? This is one of my favorite questions because I get a lot. I'm sure lots of other people do as well or thinking about it. So because of the way marijuana is classified, it's very hard to do rigorous controlled trials to see whether or not marijuana has any benefit in myotonic dystrophy. I've certainly had patients who have reported some anecdotal benefit in terms of pain or GI symptoms, but I think I myself have to be a little bit concerned, because it's a therapy that can certainly impact motivation and sleepiness, and so. For those patients who are having a lot of difficulty with motivation or getting to work or doing things, I have concerns that's going to kind of double down on that effect. But to be honest, there's really not research on the effects of marijuana in myotonic dystrophy type 1. Until it's reclassified from a Schedule 1 to Schedule 2 drug by the federal government, there's unlikely to be lots of research the way that you kind of want to see whether or not it was beneficial. Oh, here we go. Do most myotonic dystrophy research have actual DM patients? Yes. So if you're a physician like me, I'm an MD, I see patients with myotonic dystrophy, and we do research in myotonic dystrophy, and I think, I'm personally motivated to do research in myotonic dystrophy because of the patients I see in my clinic. So that is correct. And then I think, see Nadine with the hook, but will they use people from natural history studies for other studies? Our hope and obligation is that should a therapeutic trial come along, that we would help direct those people who would be eligible, at least let them know about the natural history study, or excuse me, a clinical trial, if it was to be done, if the opportunity presented itself. So yes. Oh, there's a question about how many years do we expect to have an approved treatment? Hey, that's a great question. I think, if you look at the data, it really is, 12 years. I was digging through but there are certainly examples where treatments have entered clinical trials and gone through in less than 3 or 4 years. So I think it depends an awful lot on how effective that therapy is, frankly. And there's a question about looking for a good neurologist. We should be looking at research hospitals. I think, for those neurologists who are in the myotonic dystrophy network, they're certainly familiar and experienced, and many of them participate in the development of care considerations for myotonic dystrophy. So they're very much familiar, but if you're not near one of those hospitals, then your local MDA center often has somebody who's familiar with the condition. All right, thanks everyone. I hope you had a great meeting. I had a lot of fun, and we will see you next year, hopefully in person.